The two major modes of reproduction would be asexual and sexual. So some features, characteristics about sexual and asexual modes you'll have to remember. So first of all, we'll try understanding some differences between the asexual and the sexual modes. What are some of the differences? Asexual, to write down some characteristics. Asexual, to write down some characteristics and compare with the sexual mode of reproduction. So first is, what are some modes? What are some?
Produces young ones from a single parent.
So we will write the first point is involves a single parent. Involves a single parent. Second, what about the offsprings? लिखना है. What about the offsprings? The number of offsprings are many. The process is simple. Offsprings formed are many. Offsprings formed are numerous. Offsprings formed are many. What about the offsprings? Are they the same like the parent?
No.
One parent involved, so they are all identical to the parent, no? So offsprings formed are many, and next is offsprings are identical. Offsprings are identical. Offsprings are identical. So what is the meaning? The parent and the offsprings looking alike. There's no difference at all. Next is no gametes are formed. No gametes formed. What are gametes? Gametes are specialized reproductive cells. There's no concept of gametes at all. No gametes formed at all. And then next is when we are talking about the process also, variation. Let us try understanding. Are variations there? No, variations are not there. Variations are not there, and that's the reason, or even if it is there, it is minimal. And that's the reason the offsprings are all identical. Few number of offsprings are identical. Gametes are not formed at all. Contrast, if you have to write down here the concept of sexual reproduction. What are some of the concepts of sexual reproduction then? What are some of, yeah, this chapter started in school?
Yes.
So you at least know something, so keep answering also. So sexual reproduction, when we are talking about, both parents are involved, two parents are involved. So what are you supposed to write? Two parents are involved. Two parents involved. The process of reproduction will involve two parents. Second, offsprings are many. No, it is a complex process. Sexual reproduction is a complex process, so offsprings formed will be few. Offsprings formed are few. Offsprings formed are few. So few offsprings are formed. Number 3. Offsprings are identical. No, identical, because variations will be there now. It is sexual reproduction we are talking about. So variations are observed. Variations are observed. And offsprings are not identical. Offsprings are not identical to parents. Offsprings are not identical to parents. Offsprings are not identical to parents. Next is gametes. Are gametes there? Yes. Special reproductive cells. Special reproductive cells or gametes are required or formed. Special reproductive cells or gametes are formed. Special reproductive cells or gametes are formed. Vijay? So now, when we are talking about the major differences between the sexual and the asexual, we are going to come across quite a number of the asexual modes now one after the other. Try understanding some major modes. There are many modes of asexual reproduction. We are going to study some of them. Some modes, let us understand which are some of the modes. Gametes means the specialized reproductive cells which are formed by the reproductive organs in a plant, in an animal. For example, if I tell you, in case of animals, the reproductive organs, the testes producing the sperms, the ovary producing the ovum, we learn the characteristics. Little bit with meiosis also I told you. If it is meiosis, how many cells are formed? 4 cells are formed, and those 4 cells are actually the gametes. So let us complete with the asexual mode, then again we'll have to see those concepts. Talking about the concept of asexual reproduction, that we understand that amoeba, let us first of all look into the process of gamete formation in amoeba. So now, the process of asexual reproduction in amoeba. We don't have that, I don't have that figure, so we'll have to draw this. As it is in this, all these figures that I'm going to show now, all for drawing, okay? So take out your pencils and let us start understanding these modes. All of you, take out your pencils, sharp pencils, and we'll have to understand those modes. And in this part of the chapter, there are 4 or 5 diagrams which are necessary, and we'll have to draw these parts. We'll have to be very clear about the diagram and the labeling. Let us start with the first mode. What is the first mode? We learn fission. Fission. What is the characteristic of fission? Fission means what? Fission means a parent cell, the body of the parent cell, the body of the parent, ये black भी बहुत light है. So now when we are talking about the parent cell, if it is a concept of fission, the parent cell, nucleus, cytoplasm will divide and form the new organism. Almost like a concept of mitosis. What was mitosis? Can you tell me 2 characteristics of mitosis? One parent cell divides to form 2 daughter cells. This is what is the method of reproduction now for certain unicellular organism. Almost like mitosis. The method of reproduction is same like binary fission. Let us talk about the first method. What is the first method? Binary fission. What happens in binary fission? Binary from the word 2. Now that we are talking about binary fission 2. So now let us start understanding the stages of the diagram. This is the parent amoeba. All of you will draw clear, neat diagrams. All of you start. So parent amoeba, what is the next stage? The elongation of the nucleus. That's the next stage. The nucleus will elongate. Elongation of nucleus. Elongation of the nucleus. Third is, after elongation of the nucleus, the next stage would be the constriction of the nucleus. So now, the cytoplasm will almost remain the same, but then the nuclear region, they will start showing a constriction, something this way. But by the time the nuclear constriction has happened, the cytoplasm will not remain exactly even like before. The cytoplasm also will show a groove. So now, elongation of the nucleus followed by the nuclear constriction. Elongation of the nucleus followed by the nuclear constriction. What is the meaning of constriction? Formation of a deepening on either side so that the entire nuclear content can pinch off, separate. Nuclear constriction. This will be now followed by the cytoplasmic constriction from either side, something like this. So this amoeba will almost take a shape something like this, and the nuclear division has almost completed. The nuclear division is almost completed. So this is now cytoplasmic constriction has happened. These are the stages you will have to remember. Cytoplasmic constriction. And then this will be followed by the formation of the 2 daughter nuclei. This will be followed by the two daughter amoeba, the two daughter amoeba. This is two daughter amoeba. Clear? So now that we are talking about the formation of the daughter cells, this set of diagrams is very important for you. Few diagrams I'll draw in the class, few diagrams I'll give you as homework, or we cannot draw here in the class. So now when we are talking about the binary fission, one is, is it not almost same like mitosis that we had studied, one parent cell dividing to form two daughter cells? Yes. Are these two daughter cells identical to the parent? Yes. Exactly. Are these two daughter cells showing any variation? No, negligible, no variation almost. How many daughter cells? Two. So the method is simple. The method is simple. Now, these type of organisms, now what type of organisms show binary fission? We may talk about binary fission. There are different types of binary fission also. We'll answer one question that I'll show you again. Binary fission are of different types. This is one type of binary fission where we are saying that the fission is almost horizontal. Actually, now I have drawn the diagram this way. The amoeba could have been drawn this way also to understand the concept of horizontal fission. If I draw the diagram this way, trying to understand that yes, the nuclear division has happened. The nuclear division has happened. So this actually indicates that the division has happened horizontally. But in certain cases, fission happens horizontally. In certain cases, fission happens vertically. Okay. So in certain cases, fission happens horizontally. So amoeba, we can draw the diagram this way also, not a problem. This is most common. What type of organisms show binary fission? Organisms that show binary fission, amoeba, or we can write euglena, or we can write paramecium. So these are the organisms that show binary fission. What are the organisms that show binary fission? The first one is amoeba, paramecium, and euglena. Amoeba, paramecium, euglena. These are the organisms that show binary fission. Clear? So these are all unicellular organisms, show the method of binary fission. Let us talk about the second method. What is the second method? Multiple fission. Multiple fission, the moment we talk about a concept of multiple fission, it should be very clear. Multiple fission is always taking place in organisms which are disease-causing forms. Single-celled organisms, but disease-causing forms. Does amoeba cause any disease? Euglena, paramecium, no, they are not pathogenic. They don't cause any disease. So what is common in all of them? They are non-pathogenic organisms. So you may also write this as a point of difference, that if I talk about binary fission, they are always taking place in non-pathogenic organisms, the non-disease causing unicellular organisms. Then talking about multiple fission here, multiple fission takes place where in the pathogenic organisms. Which are the ones to understand pathogenic organisms? I can say Plasmodium. Pathogenic organisms, which are the ones? Plasmodium. What is Plasmodium? Are you aware? You must have come across the word Plasmodium. Who remembers? Plasmodium cell organelle hai? Huh? Definitely you must have come across. So, in till 10th standard you have not come across this word Plasmodium. What is Plasmodium? Malarial parasite Plasmodium kabhi you have come across? No? This is the malarial parasite. Plasmodium is the malarial parasite. It is a parasite, so it is very small, single cell organism, but then it is carried by the Anopheles mosquitoes. These mosquitoes, when we, how do we get infected? These mosquitoes, they happen to, whenever they take a small amount of blood from the body of a malarial patient, they are actually taking in a large number of these unicellular organisms. And if they happen to bite a healthy individual, then they are dropping those single cell organisms into the blood of that healthy individual, and hence the healthy individual gets infected by the malarial parasite. What is the name of the malarial parasite? Plasmodium, carried by the female Anopheles mosquito. So all of you are aware that malaria is caused by the bite of Anopheles mosquitoes. But which is the parasite? The mosquito is not responsible for the disease. The parasite is what? The plasmodium. If the plasmodium is transferred, then only. How would the process of multiple fission take place here? Now this is the parent plasmodium. This is the parent plasmodium. Now this parent plasmodium with a single cell nucleus, it is a unicellular organism we are saying. This single cell will now start the process of multiple nuclear division this way. Will start multiple nuclear division like this. Can you tell me now what is the difference with this method and this? So far, here the nucleus is dividing number of times, multiple times, hence the name multiple fission. And this is how many times the nucleus is dividing? Only once. Here the nucleus is dividing only once. But that is not all. The moment the nucleus starts dividing, the cytoplasm also starts wrapping around the nucleus. I would say, what is this? What are these known as? These are known as the nuclei. What are nuclei? Plural of nucleus. So now this will be followed by, so this stage is what? This stage is multiple nuclear division. Multiple nuclear division. Multiple nuclear division. And then this will be followed by a stage much bigger, where a large number of these nuclei, they will now start wrapping around them a small amount of cytoplasm, and these are known as the new plasmodium nuclei, which are getting converted into new young cells of plasmodium. So each of these nuclei, they will be wrapped up by a small amount of cytoplasm like this. So the cytoplasm division is also in a different manner than the binary fission. The cytoplasmic the division is also not very simple, and even the cytoplasmic division is also something like this. So what will happen? The cytoplasm also starts wrapping around the nuclear region, something like this. The cytoplasm also starts wrapping around something this way, resulting in the formation of a large number of daughter nuclei. A large number of daughter nuclei. But now the number has become so many that the membrane gets torn, broken, ruptured, and the daughter nuclei are released free. So this daughter nuclei released free now. The daughter nuclei are released free. Where are they released free? If I have to understand that this is the plasmodium, where are they released free? They will be released free in the liver, in the blood. This plasmodium, where does it divide in the human body? In the liver and in the bloodstream, in the RBCs. They keep on destroying the RBCs. They keep on destroying the liver. These are the two prominent sites. So where is this multiple division happening? In the liver, in the RBCs, and destroying those tissues. And now that this large number of this daughter nuclei are released, each one of them, they will again grow, mature, and again start the process of division. So that's the reason pathogenic, these are pathogenic organisms. The process of division is very fast. Today, if the parasite has entered, in 24 hours, they have already multiplied in hundreds in the body. In another one day, they have again multiplied in thousands in the body. So immediately, that's why medication has to be taken. Medication will control their process of division, not allow the division happen, and the patient gets relief. So immediately, the relief does not happen. That's why. Even after taking medicine, it takes at least 3-4 days, quite a number of times, 7 days when the person gets relief. So now tell me, what are the two major differences between binary and multiple fission? Number one, what to write? Look at the diagram, look at the figures. What are some of the major differences to write? Number one, about the nuclear division, what can you write? Once, multiple division, multiple fission, nuclear divides a number of times. Cytoplasmic division, what can you write? for binary fission, cytoplasmic division is very evenly divided into two parts. In multiple fission, the cytoplasm, a small amount of cytoplasm wraps up around every single nuclei, resulting in the formation of the daughter nuclei. What about the types of organisms which show this method? This is generally showing in the disease-causing organisms. What are these disease-causing organisms known as? Disease-causing organisms are known as the pathogenic organisms. Multiple fission generally takes place in pathogenic organisms, and binary fission generally takes place in non-pathogenic organisms. So these are some of the differences, features to write down. What are the next methods? Fission, after fission, let us talk about the next method, that is fragmentation. The next method that we talk about is fragmentation. In what type of organisms does fragmentation take place? And how is it different from the rest of the methods? Now, fragmentation generally takes place in simple multicellular organisms, something like Spirogyra. Spirogyra is there, Obelia is there. These are organisms which show fragmentation. What is it? In this method, first of all, try understanding that how does Spirogyra look like? It is, I just now said that it is a simple multicellular organisms. It is a simple multicellular organism. Now, this simple multicellular organism, what happens is, this starts its development in a specific way. So, it will have the cytoplasm, and on top of that, it also has got some threads of some chloroplast also it has got. This is a green colored filament consisting of those plastids, just like the plants. The chloroplast, just like the plants. Now, where is it present? In the water bodies, the lake pond. In the water bodies, it is present. It's a freshwater organism. And this freshwater organism, the Spirogyra, if you happen to observe, test some amount of the water body, take a small amount of the water and put it under the microscope, few drops, we get to see that there are hundreds of those fine thread-like structures, and each of those fine thread is a Spirogyra. What is this filamentous structure known as? The filamentous structure is known as the thallus. The entire filamentous structure is known as the thallus. For an example, let me say that this organism has just 50 segments or just 50 cells in its body. I said it is a simple multicellular organism. It is not complex with an organ or system. No organ, no system, nothing it has, just few cells. It is multicellular but simple, very simple. Now, what is the mode of reproduction? Once this entire organism is fully mature, it will just automatically divide into few fragments like this. It will automatically divide, naturally, it will just divide into few fragments like this. It will just divide into few fragments like this. Now, each of these fragments is supposed to give rise to a new organism. But if the fragment has got very few number of cells, definitely that will not give rise to a new organism. What is the method of giving rise to a new organism? This part will just form few more cells and the thallus is complete. It does not have to form any tissue, organ, anything. So this is forming another few number of cells. I may say that this part has got already 15 cells. And it will form another 20-35 cells and the organism. That is it. It does not have to form any layers and tissues and organs, nothing. So just by simple cell division, the organism is formed. Draw this diagram next. Are you done with these two? Multiple fission, binary fission, take the next one up. Fragmentation. First you draw the entire thallus as I had drawn earlier, then the next stage is this. Let me just draw it again. First you will have to draw the entire thallus complete the entire filamentous structure first. You draw the entire filamentous structure first. Then you show the number of fragments. Then you show the number. In 3 stages, you'll have to draw the diagram. In 3 stages, you'll have to draw the diagram. Then the second stage, you show that the filament is divided into few segments. The thallus is divided into, the filament is divided into few segments. That you'll have to show now. Only pencil to use, and shading can be done only with the pencil. Light and dark shading to be done only with the pencil, and that is it. You don't have to use any colors to draw your diagrams. Now you show that the filament is broken down into few fragments this way. Broken down into few fragments. And then you complete the diagram, draw one more complete filament here. Draw one more complete filament here. Draw one more complete filament here, and don't draw anything for this. So draw one more complete filament with more number of segments. 1, 2, 3. Come on, all of you complete this diagram and show me fast. Draw fast. 3 diagrams. In 3 stages, you'll have to draw. The first thallus, second is the fragmentation you'll have to show with bigger. 1, 2, and 3. It will be very clearly shown. 1, 2, and 3. So what will be the third stage in the diagram? The third stage in the diagram is this is the mature organism. Once the organism is mature. When I say once the organism is mature means what? A newly formed organism, once it is mature, automatically it will divide its body into few fragments. Maybe 10, maybe 12, and now the fragments are there. Depending on that the fragments have got adequate number of cells, this will again try forming the complete thallus with the adequate number of cells, whatever it has, 40, 45, 50, whatever. Once done, again it will break up and continue. That is its simple method of growth and multiplication. All of you draw this diagram. This is how you have to draw the diagram for fragmentation. So how many new organisms formed we get to see? 3 new organisms formed, and now they are in the process of growth. So you have to start from one end of your page. Margin के पास से start करना है. 1, 2, and then 3 to show. That is enough. Draw the diagrams, all of you.
So the cells get divided equally among every segment?
No, just at random. That's why I said if there's a fragment which does not have adequate number of cells, maybe a fragment में सिर्फ 4 ही cell है, that will not grow. That will just perish away, decompose हो जाएगा. So is there any confirmed number that a spirogyra will always give rise to 6 new organisms? कोई specific number नहीं होगा, just random. कभी एक spirogyra से 8 भी बन सकता है, 6 भी बन सकता है. There is no specific number that's why. All of you complete the diagram. Only done. Pencil नहीं है. Now that we are discussing this chapter, all of you should have pencil. There's enough more diagram that you have to draw. So many more diagrams to draw.
Pages of the chapter.
4 to 5 pages, meaning by till vegetative propagation they must have done. तो आज ही खत्म हो जाएगा. Your school will go 3 days. I'll take a one hour. You finish the diagram fast. इसमें समझने की ज़्यादा कुछ concepts नहीं है. This particular methods only you have to understand. ठीक है? So this is fragmentation. Which type of organisms fragmentation? Spirogyra, Ovaria, freshwater organisms, remember. So this is Spirogyra. Now similar method will again come across. Now there is one common feature between these three. The next mode that we are going to talk about will be a bit different. What is the similar method? What is the similar concept that we are talking about here? The similar concept that we are talking about. Can you tell me by looking into the diagram, what is the similar thing in all these three? Similarity क्या है? One similarity between fission and fragmentation is it is the parent body which is dividing to form the offsprings. तो offsprings के साथ parallelly parent है. Offspring and parent is parallelly not coexisting. It is not possible that the parent and the offspring is parent. तो parent body dividing हो रहा है ना. So parent and offspring is not coexisting. तो इसको क्या लिखेंगे? Parent body and the organism is not coexisting. So we will always write this. Parental identity is lost. This is the feature that you should write for difference. Parental identity is lost. Meaning what? The parent and the offspring is not present ever together. So ये हो गया fragmentation. Now let us go to the next method. What is the next? We talk about budding. Now for budding, there are different modes of budding. There are different modes of budding. What are the different modes? The different modes of budding. For single-celled organisms, if I talk about budding, the third method we talk about is budding. Now budding can be in single-cell organisms. Budding can be in multicellular organisms and also in a plant. I talk about budding in yeast. Yeast is a single-cell organism. I talk about budding in yeast. A single-celled organism to understand. If it is a single-celled organism and we want to understand budding, then what organism to remember? It is a yeast that we have to understand. How does budding take place in a yeast? See these figures. I think you have got figures. You have got the concept of budding in yeast, but the figure is not there. So if I just talk about a concept of budding in yeast, see there, the concept of budding is the chain of buds in yeast. Now, this is a common concept that we always understand. The process of budding in yeast. What is yeast? Did you ever come across yeast? It's a fungus which is used in the bread, bakery items, no? The bread and the pastry and the buns, always yeast dala jata so that it becomes fluffy and spongy. So that's a fungus. How does it reproduce? It's a single cell. How will it reproduce? It's a single cell. And this single cell, that is the parent yeast, how will it grow? It will start dividing its nucleus and the cytoplasm in such a way that it will form, first of all, a small bud on its body. And what is happening to the nucleus? The nucleus is getting elongated and trying to divide this way, followed by the division of the nucleus. The nucleus is dividing once. The cytoplasm is dividing unequal. Binary fission ke jaise, where we had said in binary fission, what was it? Nucleus and cytoplasmic division was equal. Yahan pe nuclear division is equal, but cytoplasmic equal nahi hai. Only a small part of the cytoplasm is going there. What is this known as? This is a bud. This is a bud. Next stage, what should it be? This parent organism will be there, but the new bud will start growing. The parent is there, but then the new bud has started growing even more. By that time, the nuclear division is almost done. By that time, the nuclear division is almost done. The nucleus has separated out and the new bud is almost getting formed. This new bud will complete its growth remaining in contact with the parent organism. It will not immediately detach out. Observe, the bud is already grown. Now we understand that the bud is completely developed. Nucleus भी settle हो चुका है, यहाँ पे भी nucleus settle हो चुका है. And yes, this is the parent organism and this is the newly formed, but the new organism formed. So by rule, when the new organism is formed, it should detach from the parent body, but it is not detaching. It will not detach. What will happen rather is, it will now again give rise to one more new bud. It will give rise to one more new bud and it will now start showing the process of nuclear division. So in this process, what do we come across is that this is the parent and this is one organism, this is another organism and this is another organism. This is the parent, the next organism and this next organism and this is in the process of growth and giving rise to a new bud. This is what is known as a chain of buds in yeast. This is a common method of growth. So yeast that we get to see also when we get them, when we buy them, they are all in the form of tangled fine thread-like structures. If you put them into the microscope, they are all in that stage. All in the form of a chain. The parent organism is there, but then the parent organism immediately does not get detached with the newly formed organism. They will remain attached with the parent organism. Let us talk about one more feature, then we more or less generalize. This is single cell organism. Yeast and single cell organism. What about this? I talk about the next one. There is a hydra. Now this is a multicellular organism. Yeast unicellular organism tha. Now I talk about a multicellular organism. So multicellular mein how can budding happen? In a multicellular organism, if you have to understand what example to write, hydra. Majorly, there are so many cylindrates present again. These are present in the shallow water bodies, right? The river, the lake, the pond, and these are also fine thread-like structures. We can just see them with our naked eyes, but we cannot see the details. For seeing the details, we need a microscope. So this single-celled organism, the hydra, this single-celled organism, how does it reproduce? Method वही same है. But यहाँ पे, since it's a multicellular organism, it can give rise to many buds at the same time. This hydra also does not have well-developed organs and systems. Few tissues here, but it can give rise to many buds at the same time. Whatever it is, 5 buds hai, 6 buds hai, whatever it is, buds means what? Accumulation of some fast dividing cells. What is a bud? An accumulation of a rapidly dividing cells, fast dividing cells. So this hydra also will give rise to a large number of buds. What is the meaning of the word buds? In certain regions of the body, there will be an accumulation of fast dividing cells. And the cells will divide fast. Process of growth is rapid. Okay, so process of growth is happening. The bud is growing, the new parts of the body developing, the body, the tentacles and everything developing. Once it is fully developed, till the time it is fully developed, it will remain attached to the parent body. Once fully developed, then only it will detach from the parent body and then grow and develop the optimum size and again start forming buds. So I say the number of buds specific kuch nahi hai. Is it anything confirmed that one hydra will give rise to 5 buds and 5 new organisms? Nothing confirmed. What about the number of offspring formed during the process of budding in hydra? It is variable. What I supposed to write as an answer? The number is variable because the number of offspring formed depends on how many buds will be formed. कभी कभार 10 buds में से 4 buds 5 buds will start developing into an organism. So how many buds will the parent form and then start the process of growth? Complete होगा, पूरा growth complete होगा, remaining in attachment with the parent body, then get detached out, and then it will grow, complete its size, and फिर से process of bud formation शुरू हो जाएगा. Till here is it clear? We have studied binary fission, multiple fission, fragmentation, budding in yeast and budding in hydra. एक और concept of budding, that is the budding in bryophyllum. But this is actually a concept in plants. This is also known as budding. This is also known as budding. इसमें क्या होता है? This is a type of a plant, bryophyllum. Now, these plants are not very tall plants. These are common plants that we come across in the gardens and all. बहुत common plants, But these plants, if I talk about the bryophyllum, how tall are these plants? No, these plants are not very tall. And once the leaf is broad, the leaf is big, broad leaves they have. And once the leaf is broad, what will happen is the leaf will always give rise to some small buds. All of you must have seen some buds of those potatoes. देखा ना? Those buds are the almost same like that, but green in color. तो इस तरह के leaf के margin में एक buds develop हो जाता है काफी सारे. And then from here, some fine root-like structures also starts forming. So after some time, when this leaf falls off, and all these buds, along with the buds and the root hairs, the leaf falls off with the bud and on the soil. So these secondary roots coming out from the buds, ये secondary roots को क्या बोलेंगे? These roots are known as, please take down these words when you frame your answers to use. These are known as adventitious roots of the bryophyllum. तो ये देखो adventitious roots, these are known as the adventitious roots. So ये adventitious roots come in contact with the soil, and then in a week time what will happen? The leaf will all perish away. ये पूरा decompose होके mix हो जाएगा soil के साथ, and then new plants will develop. आया समझ में? So a type of a plant which shows bryophyllum, which is a plant which shows a type of a plant which shows budding, bryophyllum. Type of a plant which shows budding bryophyllum. So what is the method I told you? Once the leaf is fully grown, some buds will develop on its margin, followed by some fine hair-like structures, root-like structures. What are these root-like structures called? Adventitious roots. So काफी बार, since the plants are short, काफी बार ये the leaf is with the plant attached, and the roots have already. No, just like those money plant, those roots. Did you see the ones old buildings also? Very common these plants are. So even in case of money plant, did you see that the long roots keep hanging? So almost like that, money plant के roots के जैसे इतना thick नहीं है, but these are fine fibrous, and this will touch the soil. Even when the leaf is in contact with the parent plant, या तो कभी-कभार, most of the time when the leaf falls off. Another buds coming in contact with the soil, the roots coming in contact with the soil, and then the growth of a new plant. This is the concept of budding. We just take a few more minutes, we'll complete with this concept of asexual reproduction, all of it. The next is the spore formation. Where does spore formation happen? Spore formation happens in fungus. Now that this monsoon month, now that we are talking about the monsoon month, all of you must be aware that if you take out some leather items from your cupboards here or there, you must have come across some white granular structures, and there is a characteristic damp smell also there. What are these? These are the rhizopus. So we talk about the next type of method, the next method, that is the spore formation. Spore formation a method which takes place in fungus, in all fungi. Spore formation is a method which takes place in the fungi. Name some of them. Which fungi? The rhizopus. How many of you have seen the bread mold? Bread के ऊपर mold. How many of you have seen? देखा कभी? Bread के ऊपर mold. That's very common, no? So on the surface of the bread, 2-4 दिन अगर हम bread packets को ऐसे ही रख देंगे out from the fridge, So we get to see in this moist weather, it is very common. Fine cottony growth develop हो जाता है. So how does this method happen? In what type of organisms? Remember, rhizopus, जो common fungus है on the leather items, bags में, shoes में, cupboards में. Food items में, bread mold on the citrus fruits also, orange में, sweet lime में, and the bread में, pastry में, all these moist food items. How does the method happen? The method is very simple, it's similar like multiple fission. First of all, this structure, the fine filamentous structure, multicellular. The fine filamentous structure, multicellular. What are these structures known as? Take the word, hypha. ये जो diagram है, hydra, you will draw, space रखो notebook में, half a page, draw the diagram of hydra. This diagram also you will draw nice, but just abhi samjho. So hypha. Then what will happen? These are multicellular structures. Only one single cell, it will undergo the process of multiple nuclear division like this. Multiple nuclear division like this. And maybe in one of them, the multiple nuclear division has happened so many that the membrane of the cell already got torn and the spores are all free. This is the method. So now, one of these cells, they have, each one of them, they had got one cell, and one of them cell, they underwent multiple division a number of times and resulted in the formation of a knob-like structure. What is this knob-like structure known as? This is known as the sporangium. Very important, this word always comes in the exam. What is the knob-like structure formed in rhizopus known as? What is the knob-like structure known as? Sporangium. And what is the filament? All of you must have seen bread mold में tiny dot-like structure रहता है and then fine fiber thread. वो thread को क्या बोलेंगे? This is known as the sporangiophore. Sporangium and filament is sporangiophore. Take a note of these two words, very important. Name the knob-like structure and the filament. Filament, sporangiophore, knob-like structure, sporangium. So, and what are these? Fine granular structures, these are spores. ये diagram अच्छी तरह से draw करके next class में मुझे दिखाना. So, this particular method, the fungal reproduction, then these spores are released. The spores are very minute, very small, light. They can be blown away far distances. And that is the reason the fungal infection is so very rapid. They spread very fast. So the fungal infection, even on our scalp, if I talk about dandruff, almost the same method. So fast it is. On the skin, on the animal body, always very fast. So these methods are the true asexual methods. So these asexual methods, I think they must have already finished. So till here you study well. What are the methods? Two types of fission. Fragmentation, three types of budding I discussed, and spore formation. These are the actual asexual modes. The next class will take up with the help of the plant parts how plants can grow. That is different. That is vegetative propagation. Okay. So you study till this month. Regeneration is a different. Regeneration asexual mode nahi hai. This I will take later with vegetative. Asexual mode is only this part. Regeneration is just a method to repair the lost part. वो typical method of reproduction नहीं है.
Two ठीक है भाई।
डेढ़ है भैया, आपको नहीं पता क्या बात कर रहे हो?
अच्छे भी मार दो।
क्या बात
क्या
कर रहे हो?
सही में मार दिया
तो
ये गलत बात होती है यार।
मैं नहीं देखा हूँ। गलत बात होती है तो तू सही कर रही है, बता। उसको बोल टाइम क्या होगा।
अच्छा तो मुस्लिम वाले लोग रैली निकाल